Like I just said, thanks, Andreas, very much for the introduction. And also thanks to everyone for making the time to join us today for this award ceremony. I know very much, I would have loved to have the in-person meeting, but yes, the current situation calls for new measures, and this is the new way to go. I would give let's say a small presentation on my doctoral research on memory B cells, which I conducted at the Deutsche Roma-Forschungszentrum in the group of Professor Radburg. And B cells in general are one of the defensive immune cells we have, and they do this by producing what we call the antibodies. Antibodies are very important in the clearing of foreign pathogens like viruses and bacteria. And usually without this line of evidence, the body is not able to fight most infections. So in principle, when the immune system encounters a foreign pathogen or antigen, a small fraction of the naive B cells, which are reactive to this pathogen, undergo what is called the clonal expansion to produce effector cells, which will then mount the defense against the foreign pathogen. At the very peak of the immune response, some of these B cells undergo what is called the class switching and also the affinity maturation of their B cell receptor. In so doing, they come out with a new isotype phenotype and also a B cell receptor, which is much better fine-tuned in terms of affinity and specificity for this pathogen. As the foreign pathogen is cleared from the system, most of the B cells die out via apoptosis, leaving behind a small fraction of antigen-experienced long-living B cells, which we term the memory B cells. So in the event that the immune system re-encounters the same or similar antigen, it is now equipped to react much faster and better to the antigen. And this is the underlying principle of what we call the immunological B cell memory. So my doctoral research focused on the understanding about the heterogeneity, the distribution, lifestyle maintenance of tissue resident isotype switch memory B cells using mouse models. And this is what I'm going to share with you some of the findings that I got during my doctoral research. Usually in the lab, to study memory B cells, what we do is to immunize mice at consecutive time points and then wait whilst the immune response has gone down in the memory phase. And then we could analyze the memory compartment. We can then identify the memory B cells using a combination of positive and negative markers that we stain at the protein level using FACS here. And here it's shown as an example for IgG2B as an isotype of memory B cells. So the data that I'm going to show you today is basically based on the same gating principle for identifying the isotypes rich memory B cells. One of the first observation that we had was using different immunization protocol with different antigen, different routes of immunization. We could observe that as it has previously been described, that the spleen is seems to be the main organ for the maintenance of isotypes rich memory B cells, here shown for IgA, IgG1, IgG2B. But one could also appreciate the fact that the bone marrow maintains a decent proportion of these isotype switch memory B cells, much higher than in the other lymphatic organs or in the peripheral blood. So to say that here, the two dominant organs for the maintenance of isotype switch memory B cells are the spleen and the bone marrow. One of the key features of memory cells in general is that these cells are quiescent and resting in terms of activation and proliferation. So we wanted to know whether in these cells we are looking from the tissues, the bone marrow and the spleen, share this memory phenotype. What we did was to stain with Ki67, which is a transcription factor expressed mostly in proliferative cells, but not in quiescent cells like the memory cells. And as you can see here, indeed, most of the cells that we identified in the bone marrow and the spleen do not express Ki67, thereby confirming that indeed these are quiescent cells. This is in sharp contrast to the high expression of Ki67 in the much proliferative compartment like the germinal center B cells in the spleen. We could also confirm this observation by treating immunized mice with cyclophosphamide, which is a therapeutic agent used in the clinic mostly to target highly proliferative cells like tumor cells. And as you can see here, indeed, such a treatment causes a drastic reduction in the entire B cell population, here represented by CD19. But if you would then look at the isotype-rich memory B cells, for example here, IgG2b, there is no significant reduction of the memory compartment. So these two lines of evidence clearly show that the tissue memory B cells that we are talking of here are of the quiescent and resting in terms of activation or proliferation. So as curious scientists, always the question is, these memory cells that we're looking at from the bone marrow and the spleen, are they the same or are they different? And what are some of the characteristics that we can use to describe these two compartments? One of the ways we chose to address this question is looking at the entirety of the B cell receptor repertoire, or more to say, the antibody repertoire at this stage at the bulk population level. And what we did is to isolate memory B cells from the bone marrow and the spleen, and then at the clonotypes level, compare whether the antibody repertoire uh, is the same or are different. So at the first glance, if you compare the number of the clonotypes of antibodies in the bone marrow and the spleen, you could observe that there's only a marginal overlap between the bone marrow and the spleen. So at the first glance, this line of evidence suggested to us that memory cells of the bone marrow and the spleen could represent two distinct populations of memory B cells. Furthermore, we looked at how the B cell receptor or the antibodies of these two compartments are made of. And we looked at the hemoglobin heavy chain segment, which is of the variable region that the antibodies use to identify foreign antigens or pathogens. And as you can see here, between the bone marrow and the spleen, there seems to be a significant difference in what types of hemoglobin heavy chain segments are used in the formation of the antibodies. This is in the fact that some of the IgA genes are exclusively used in one organ but not the other, or that the relative proportion differs between the two compartments. So at this point, the evidence at the bulk population level, from the clonotype level, and also how the antibodies are made, suggests that these two compartments of the spleen and the bone marrow represents different populations of isotype-rich memory B cells. So coming to a topic which also Leonard mentioned on, we took advantage of the novelty of single cell sequencing to set up a whole pipeline of tools and analysis to investigate the heterogeneity of these memory B cells further. So here we perform single cell sequencing of several thousands of individual and isotype-rich memory B cells from the bone marrow and the spleen, and not only looking at the transcriptomics, but also combining this with the B cell receptor analysis. As shown here, using clustering analysis, we could identify distinct clusters, or more to say subpopulation of isotype-rich memory B cells, including ones which had previously not been described. If you would also look in closer, then the differences between the memory B cells in the bone marrow and the spleen becomes quite evident. in that you can see here that some of the subpopulation of the isotype switch memory B cells are exclusively in one organ but not the other. So for example, here the cluster 5 and the cluster 4 are either in the bone marrow or in the spleen. Furthermore, if you look at the gene expression profile of these different clusters, you can see that they really differ in the genes which are upregulated or downregulated in this compartment. So the two lines of evidence at the gene expression level from the bulk population analysis and also from the single cell sequencing clearly shows that the memory B cells of the bone marrow and the spleen differ differ in the size of the clonotypes, also differ in how the B cell receptor is made and also differ in the composition of the subpopulations in the two organs. We wanted to then verify this observation from the gene expression analysis. So what we did is to take some of the differential genes that we found from the transcriptomes and see whether we could confirm this observation at the protein level. And indeed, this was what we could find at the protein level using facts that the two populations that we saw from the uh, transcriptome analysis were indeed evident also at the protein level, in that they were either in one organ the but not the other. Just to give a small insight of what are some of the differences between these two compartments. So the cluster 4, as I showed previously, this was exclusively in the spleen, and the cluster 5 was exclusive in the bone marrow. One of the differential genes was the expression of CD21 and CD23, which differed in expression between the two compartments. Also, furthermore, these two clusters differed in the genes which were highly expressed or down expressed in here. One thing of interest is that if you look at the bone marrow compartment, these were more of more quiescent phenotype, in that they do not express genes that one would attribute to activation or proliferation. And also, if you would then look at some of the key characteristics in terms of what kind of the antibodies and how exclusive they are. For example, the cluster 4 here, this is completely different from uh, all the other 5 subpopulation of the isotype switch memory B cells, which I earlier showed on. So indeed, the memory B cells of the bone marrow and the spleen have what more or less to say an exclusive population of memory B cells, which kind of probably might also give them different functional implications, which we do not know as of now. So the evidence that we've talked about, starting from the enumeration of memory B cells throughout the bulk population analysis, through the single cell sequencing, clearly shows that the spleen and the bone marrow are the two major organs for the maintenance of isotype switch memory B cells. And that if you would zoom in further, the memory B cells of the bone marrow and spleen represent two distinct subpopulation of tissue-resident memory B cells. So now we shifted our attention to the bone marrow, which was more of an organ which is quite of interest to us, also from the immunological memory point of view. So here we use histology studies to look at the distribution of memory B cells in the bone marrow, staining for various sections. And as shown here, memory B cells are identified by the red staining here, IgG2b, as an example, were evenly distributed throughout the bone marrow without any preferential localization or clustering. As we zoomed in further using co-stain analysis, staining for markers known to represent other immune cell types, one of the striking striking observation was that most of the isotype switch memory B cells were located either directly or in close proximity to stromal cells, here represented by VCAM1 expression, fibronectin, or laminin. And what I must say is that this phenomenon currently seems it's not unique for memory B cells, but it has also been described for other memory cell types like memory CD4, CD8, and also plasma cells. Currently, we believe that this close interaction between the memory B cells and the stromal cells could be explained by the fact that the memory B cells have receptors, in this case VLA4 and VLA6, which are then attracted to the ligands expressed by the mesenchymal stromal cells. So there is what we call in the field as the stromal niches for the memory B cells. As I just mentioned, the evidence also from other immune memory cell types like CD4, CD8, and plasma cells strongly suggests that there's a role of stromal cells in the maintenance of immune memory. So this was one of the key interests that we also had. It was quite challenging, but we also managed to set up a lot of methods that allowed us to look in further into the biological and functional roles of stromal cells. We, after a long trial, we were able to set up a method that allowed us to isolate directly ex vivo bone marrow mesenchymal stromal cells for analysis. So here, once again, and together with the whole team, we managed to set up the single cell sequencing of stromal cells to then look in more at the functional and the biological properties of these cells. I wouldn't go so much into details about all the analysis of this, but one of the key messages is that from the sequence analysis, we observed a quite enormous heterogeneity of mesenchymal stromal cells, and this initially precluded the identification of unique subpopulations. But then we were more interested in looking at how this interaction between the memory cells and the stromal cells were. So we pre-selected over 100 genes known to code for cytokines and chemokines relevant for the development, maintenance, of survival of various immune cells, and then perform a further clustering analysis. In this way, we could identify 16 distinct subpopulation of bone marrow mesenchymal stromal cells, which are unique in that they express either only one of a type of a chemokine or cytokine, but not the other. This is in sharp contrast to a lot of other cytokines or ligands like the midkine and laminin beta 1, which were expressed ubiquitously all over. And of quite interest is the fact that we also, more or less to say, at a more pronounced level, identify a subpopulation of bone marrow stromal cells which express above key cytokines and described for the maintenance of B cell in general. So this information here now would serve as further stone for investigation into the role of the various cytokines and chemokines produced by stromal cells for the maintenance of immune memory cell types. So if I would give you one message to take home, is that the findings of our research show that the bone marrow maintains subpopulations of quiescent isotype switch memory B cells, which have distinct antibody repertoire, at least in this case from their compartment in the spleen, and that these memory B cells are maintained in stromal niches, probably via receptor ligand interactions. And yes, basic science is interesting, curious minds as we are as basic scientists, but also as someone with an ND background, I'm thinking of how we can translate this from the bed to the bedside, which is something that Leonard also touched on. So with the tools that we've kind of developed, which allows us to look at distinct, look at individual cells at single cell resolution, this kind of gives us a tool that we can also bring or translate into developing therapies. In this way, it would be possible to uh, furthermore address the heterogeneity of memory B cells in the settings of various chronic inflammation or rheumatic or autoimmune diseases. And one of the ways would be to identifying and then targeting the specific subset of pathogenic tissue resident memory B cells. For example, as Leonard touched on, that if you could find a unique marker, like let's say CD38, then you could have therapies that you could direct specifically to this subpopulation whilst minimizing the side effects that you would encounter from other therapies. The other way would be the indirect way, where you would focus on the stromal cells, which are organizing the survival niches for these pathogenic memory B cells. So it's our hope that these findings will not be only in the field of basic research, but also find its way to developing therapies that will bring relief to patients all around the world. I yes, I am the one here today giving a presentation, but I must say it has been more than a teamwork. It's been uh, 4 solid years of developing new tools, new methodologies, a lot of scientific discussions, hours of discussions, just to get an understanding of how we can get the best out of science. And I would say a very sincere appreciations to Andreas for let me put it in the football terms for scouting me out and then having the belief in me that even though I didn't have so much of basic research as a physician, he could entrust me and believe me that I could deliver in also in a scientific career. Also, a whole thank you to the whole group of Radbo, to uh, Professor Chang and Mashiriki for the lots of hours of discussion, and especially with the single cell sequencing, the whole team for Renee, with whom I would say this project we spend the most of the time working on, and all the folks from the sequencing department and the other group members of the Radbook, I would say the Radbook dynasty. So the findings of what I shared with you are both published and last the year before and this year, and one can also get access to further read more on this. And with that, I would say a very big thank you for giving me and Leonard this award, and also for your attention today to listen to us. Thank you so much.